I'm Per Johnson, and this is the effects of temperature on learning complex tasks in red harvester ants. Global warming poses a severe threat to nearly every organism on the planet. Rising temperatures are projected to have drastic ecological consequences on every trophic level. This study focused on the behavioral changes that come about as a result of these higher temperatures in one of the world's smaller species, the ant. Ants are one of the most intelligent families of insects. They belong to a group of insects referred to as eusocial, indicating their aptitude for interaction and communication, as well as their capacity for cooperation. While other insects, like various species of bees and wasps, are also known as the most intelligent insects, ants' ability to form multi-generational colonies with hierarchical division of labor demonstrates just how advanced they are. Ants are also highly beneficial to the environment, performing soil aeration through their natural tunneling behavior. Climate change will threaten ants in the near future through habitat and food shortages, which could have implications for plants that rely on soil aeration to grow and repopulate. To learn more about how organisms are able to overcome obstacles, researchers investigate behavioral flexibility, a cognitive tool that allows animals to make changes in their behavior in response to the surrounding environment. This property has been extensively studied in other eusocial insects to examine their capacity for tool learning, with research done on bees learning to push balls, pull strings, and select a path based on colors and patterns. However, beyond a study proving ants can learn to use tools to pick up unfamiliar objects, little has been done to evaluate behavioral flexibility in ants. This study sought to fill that gap, as well as to contextualize adaptive behavior in a critically relevant context. This experiment had two major objectives. First, to verify that ants could learn a complex challenge similar to the ones done in previous studies with other insects, and second, to determine the relationship between temperature and behavioral flexibility for that challenge. The task that ants were tested on was based on the color selection experiment performed with bees. A Y-shaped chamber was designed using Fusion 360 software and printed on a Prusa 3D printer. It was designed to be a binary choice with a distinct color on either side, a target color and a non-target color. Ants were tested on their ability to learn to recognize the target color and repeatedly select it in a test. The experiment was performed multiple times at different temperatures to evaluate the ants' visual cognition in a warmed environment. The temperature of each chamber was the independent variable, and the time to complete as well as success of each trial was the dependent variable. The hypothesis of the experiment was that the ants tested in the warmer conditions would be less able to learn complex tasks and would perform worse on the Y-chamber test than ants in room temperature conditions. Over the course of the study, over 200 ants were in the laboratory to be tested. They were stored in small plastic habitats with holes for airflow in groups of about 20. Each habitat had 4 centimeters of sand for tunneling and burrowing so the ants could behave as naturally as possible while awaiting testing. Both a dry seed mix and minced fresh cucumber were given to the ants every other day for food, and a wet cotton ball was placed in the center of their chamber for moisture and hydration. All ant groups were stored at 23 degrees Celsius until the experiment began. To test the behavioral flexibility of the ants, they were individually placed inside the choice chamber. Ants are trichromatic, meaning they see in blue, green, and ultraviolet. 2 distinct shades of blue and green were selected for the choice colors, and blue was arbitrarily chosen to be the target color for the experiment. 3 groups were created, a control group, a 23 degrees Celsius test group, and a 36 degrees Celsius group. Red harvester ants come from the southwest United States, so 36 degrees Celsius was selected to provide an extreme temperature from their specific environment. The outline of each trial was similar across each group. With the exception of the control group, each ant was first placed inside a small exposure chamber with a blue paper circle and a drop of 30% sucrose water solution. Each time they entered this chamber was 1 exposure. After being allowed to investigate the water in the circle, they would be placed inside the Y chamber and timed until they selected a side of the maze. If the ant selected the blue side, it would be rewarded with a drop of sucrose water. If it selected the green side, it would not. Regardless of selection, each ant was then placed back inside of the exposure chamber and shown the blue circle and sucrose water again, restarting the process. 3 ants were tested at each temperature, and this method was performed with each ant 5 times. The only variation between groups was that the control group was not given any exposures to get baseline data about whether the exposures actually taught the ants anything, and the 36 Celsius group was tested in an incubator at 36 degrees Celsius after being warmed for 72 hours prior to experimentation. After testing had been completed, results were processed using statistical analysis. In response to the first objective of the experiment, determining whether the ants could learn this task, choices of the 23 degrees Celsius group were evaluated. Using a chi-square test, the ants were found to not significantly select the target color. However, the p-value of the test was 0.058, which is incredibly close to the target p-value of 0.05 that would indicate statistical significance. The second objective was to determine the relationship between behavioral flexibility and temperature. Through a second chi-square test, the 36 degrees Celsius group was determined to be even less significantly able to perform the Y-chamber test. Comparing the 36 degrees Celsius and the 23 degrees Celsius groups using a t-test, the 23 degrees Celsius group was found to be significantly better at correctly completing the maze than the 36 degrees Celsius group. In addition to the success of each trial, the time to complete each trial was measured for each ant. There was no relationship between number of exposures and the time it took for the ant to solve the maze, regardless of which color they selected. These results can be condensed into three broad statements. First, ants in the 23 degrees Celsius group did not significantly select blue over green in the choice chamber. Second, ants in the 23 degrees Celsius group did choose the correct path significantly more than both the 36 degrees Celsius and the control group. Third, there was no relationship between the number of exposures and the time to complete across the individual ants. The overall hypothesis of the experiment that ants tested in warmer conditions would be less successful on the Y-chamber test than the ants tested at room temperature was supported. Even though the data didn't quite statistically prove that ants could learn this color selection task, the low p-value demonstrates that the ants were on trend to perform the task successfully. Time constraints prevented additional testing, but a larger dataset could increase the statistical significance of the study. Previous research also shows that ants require several trials to visually learn new information, verifying the potential of an expanded trial size. Beyond just increasing the sample size of the data, future expansions of the study could include a more complex chamber, as well as additional temperatures to determine at what point visual cognition is severely influenced by temperature. Although the results were not definitively statistically significant, the study sets the groundwork for future experiments in ants' cognitive abilities and their limitations. This study also proves a decrease in behavioral flexibility for ants as temperatures increase, which poses a threat to all eusocial insects and dependent organisms in their ecosystems as the Earth's temperature rises.